Bon dia a tothom. Ara començarem la primera taula, que porta com a títol Explorant el deteriorament cognitiu lleu, biomarcadors i factors predictors. I jo el que volia fer primer de tot i de forma molt breu és agrair a la comitè organitzador de la Societat Catalana i a la Facultat de Psicologia de la Blanquerna l'haver organitzat aquesta taula. Penso que jo estic molt content i és un honor per mi poder moderar-la perquè realment hi ha ponents d'un alt nivell, com deia abans la presidenta, la Maite. Quan vaig parlar amb la Maite i la Sílvia per fer aquesta taula, la idea era qui és el millor que fa això, i realment això és alguna cosa que ara està dirigint l'esperit de la societat i estem molt contents perquè el nivell augmenta i augmenta. Simplement agrair poder tenir els ponents, que ara introduiré breument. First of all, thanks Renaud, Michael and Albert for accepting this invitation. Very pleased to have you here in Barcelona and looking forward to hear your interventions. Primer de tot, introduiré per ordre del que exposaran. El doctor Renaud la Joie és doctor en neuropsicologia, va fer estudis de doctorat a la Universitat de Caen, a França, i també a la Universitat de Califòrnia a Berkeley, amb el doctor William Jagust. Actualment treballa a Lyon, a cavall entre Lyon i Caen, a França, com a investigador postdoctoral, i realment té una línia d'investigació, està treballant molt amb la imatge, amb la malaltia d'Alzheimer i l'envelliment, els correlats cognitius i els aspectes de neuroimatge que relacionen amb biomarcadors, i el podem veure que és una persona molt jove, però realment té una productivitat i unes publicacions impressionants a les revistes de primer ordre, Brain, Neuron, etc., i un impacte molt gran amb el que està produint actualment, i per tant serà fantàstic poder-lo sentir.
Good morning to everyone, and I'd like to thank everyone for being here and the organizers for asking a very young researcher to come here and talk to you about Alzheimer's disease. Basically, the presentation I'm going to give is going to be a review of the research over the last few years, because there have been a lot of works on the topic of neuroimaging biomarkers of Alzheimer's disease and how they could be used and what they could mean in cognitively normal elder adults. So I'm just going to give you a quick introduction of Alzheimer's disease, because I think most of you are very familiar with it. And after that, I'm going to focus on those neuroimaging biomarkers, trying to explain which one I'm going to talk about, because there's many of them. And I'm going to try to develop their relationship to cognition in elder people. So basically, this is always very complicated to do when you try to define Alzheimer's disease when you give a talk, because the more and more I work in the field, the less I'm really sure what I'm talking about. So I'm going to give you the classical definition. So basically it's a dementia, so cognitive deficits that interfere with daily function. And in the context of Alzheimer's disease, it is expected to have to start very slowly, to have a very insidious onset and slow or at least gradual progression. And it's usually expected to start with episodic memory deficits, even if there are some exceptions to this. And on the other side, there is neuropathological alterations that are included in the Alzheimer's disease definition. So basically, you can group them in three different alterations. So tangles, which are abnormalities within neurons, and they're made of tau protein. You've got beta amyloid peptides that form plaques outside the neurons, and then more macroscopic neuronal and spinal synaptic loss. So the classical definition of Alzheimer's disease is this clinical pathological entity. But usually in everyday clinic, when you diagnose Alzheimer's disease, you basically do it only on the clinical presentation. And it's, properly speaking, probabilistic clinical diagnosis. Because the definite diagnosis for Alzheimer's disease is only when you can show those two features. So it's only post-mortem. And the problem with Alzheimer's disease is that the overlap between symptoms and pathology is not very clear. And if you try to represent them here, in the middle, you have definite Alzheimer's disease, where you have both the symptoms and the pathology, but you can also have people that have one or the other. And the overlap is actually not so good. You can see here in the meta-analysis, that is a little bit old now, but you can see that the clinical diagnosis does not allow to identify pathology with a lot of precision. The other problem with the clinical symptomatology of Alzheimer's disease is that the clinical diagnosis is pretty late in the disease process. So here you have a very interesting study. It was an epidemiological study of almost 4,000 people that were followed up to 14 years, and some of them developed Alzheimer's disease. They were matched to controls that were still healthy, and you can see here, and I really like this chart because it's very striking, I think. Differences were, in terms of group differences, of course, were identified 8 years before the clinical diagnosis on the MMSE scale, which is, as you all know, very not very precise and sensitive. And you can even use more sensitive tests like verbal fluency or assess cognitive complaint, and you can reach similar charts. So there's a very long phase during which the disease progresses, and it's not only brain changes. You can see cognition itself also changes way before dementia can be diagnosed. So I guess you're also very familiar with the concept of mild cognitive impairment. That is a clinical entity that is expected to capture those pre-dementia phases of Alzheimer's disease. So it's really at the transition between healthy cognition and dementia. The problem with mild cognitive impairment is that it's a very heterogeneous group. So some of them will develop dementia, but some of them will remain stable. Some will even return to normal cognition, and some of them can convert to other dementia. So the clinic itself of mild cognitive impairment is not sufficient to identify those patients who are going to develop dementia. And similar to the concept of mild cognitive impairment that was 15 years ago, now we're thinking of identifying those people with healthy cognition who are to develop cognitive impairment. So it's even going further back in the timeline of disease progression. And this is where the biomarkers can play a role. So actually biomarkers can be useful or used for many aspects. They can be used to, for example, in patients with dementia, to confirm that the symptoms that you can identify in the patients are actually due to underlying Alzheimer's pathology. They can be used to predict, they could be used to predict cognitive decline, but for a more fundamental point of view, you could use them in healthy individual and patients to study the mechanism of Alzheimer's disease. And what I'm going to present is kind of a mix of all of this. So we're always talking about biomarkers and neuroimaging biomarkers. Actually, there are plenty of them. So first of all, when you talk about Alzheimer's disease, I'm going to focus on the neuroimaging biomarkers, but you could also, and that would be another presentation actually, talk about the biomarkers you can have in the CSF, but I'm really not going to talk about this at all. And when you look at neuroimaging, there's many of them. So this is not even exhaustive, and I'm really not going to talk about this, about all of this. I'm going to talk about the three main biomarkers, or at least the three that are the most used in studies at the moment. So first of all, it's like the classical T1 weighted structural MRI. Here you have an example for an healthy subject and patient with Alzheimer's disease. And this is a group analysis of Alzheimer's disease patients. So this is the region in orange where you can identify significant and pretty strong atrophy in those patients. And as you might know, it's mainly in the medial temporal lobe, but also in the posterior temporal and parietal neocortex. So you can use this T1 weighted MRI to identify atrophy in the patients. You can use PET imaging with FDG. So FDG is basically only like glucose, radioactive glucose, and you can use it to see those regions of the brain that are the most metabolic and the most active. And here you have again two examples and a group analysis, and you can see it's pretty similar to atrophy, but we're going to talk a little bit more about this later. And the third, actually it's the newest of all three, but maybe the most popular at the moment. It's PET imaging with different tracers that can bind to beta amyloid. So I told you before, beta amyloid is one of the neuropathological features of Alzheimer's disease. And for a decade now, we've been able to see beta amyloid deposits in the brain of living patients. So the ability of seeing beta amyloid in the brain of living patients is very interesting for a more fundamental point of view, because we actually still don't know what causes Alzheimer's disease, but the main hypothesis is the amyloid cascade hypothesis, which stipulates that beta amyloid causes Alzheimer's disease. It triggers a cascade of pathological events that will lead to neurodegeneration here and dementia here. So you can use biomarkers to assess different different points of this hypothesis. You have amyloid imaging here at the very top of this cascade. MRI and FDG PET can be used to assess neurodegeneration that is supposed to be at the bottom here. And then, of course, cognitive tests to assess cognitive impairment and dementia. And you've probably seen a lot of curves. If you've read a little bit about Alzheimer's disease, I actually started collecting all different curves at the very beginning, and I think I stopped a year and a half ago because it was too big. There were too many of them. So I didn't really know which one I would present today, so I kind of made a mix of them, and this is basically what you can find, even though some of them have subtle differences. But basically, this model of biomarkers in humans is actually the translation, more or less, of this amyloid cascade hypothesis. It stipulates that amyloid here starts very early in people with no cognitive impairments. And then you can find biomarkers of a neurodegeneration here, and memory and clinical function that start later in the disease process. And a few years ago, there was a few papers that were published that were proposing a new criteria for Alzheimer's disease. And those criteria included biomarkers. And what I'm going to focus on today is those criteria for preclinical Alzheimer's disease. So it's divided in a few stages. As you can see here, you would have stage 0, which is actually non-Alzheimer's pathway, where you have no amyloid, no degeneration, you have all negative biomarkers. Then you have stage 1, where you can see amyloid deposition in the brain of people, but nothing else. That would be stage 1. Stage 2 is a little bit later in the disease process, and then you would have neurodegeneration here, in addition to amyloid. And the third stage, as you can see here, is just like stage 2 with subtle cognitive impairment. So that term is pretty complicated to implement in a study, because cognition here is still supposed to be in the normal range. You're not MCI, you're just before MCI. So this subtle cognitive impairment is pretty hard to implement in a study. So before showing you data on those individual who are positive for biomarkers, I just want to make a few points. I don't want you to get bored with technical details, but I think they really matter, and I want to convince you that determining biomarker positivity is not that easy, and it might be more complicated than what we usually think. So first of all, you have many ways to determine positivity. You can look at images, just look at them, visual read. This is usually what is done in clinics. And you can use more complicated computer-based tools to quantify atrophy, for example. And you can also look at different regions of the brain, and different regions might tell you different aspects. So, for example, you can you have visual you have scales for assessing hippocampal atrophy here. The skeleton scale is pretty common. But you have another one that can be used for posterior cortex. So maybe those people that are going to be positive on this one are not going to be on the other one. Then you can use computer to quantify different biomarkers. For example, here, those are the regions in 3 different papers that were used to assess hypometabolism in patients. And you can see they're pretty similar, but they're not exactly the same. And it might seem a bit crazy to insist on this, but actually it influences a lot the results. And the third point is, I think it's very understandable, and it's very similar to classical cognitive tests. Because actually, when you do biomarkers, just like when you do a memory test, you have continuous variables. And you have to decide of a threshold to say whether this patient has impairment or not. So of course, you know, as clinicians, that it's not that simple, but usually you're expected to give an advice and your opinion on a patient, and you're asked whether this patient has impairment or not. And you can see that the variables are very continuous, and there are overlap between patients here and controls. So you have to make compromise, and of course this is not always easy to determine. And this is really not easy, and I just wanted to show you this paper. So actually there's just two sentences that you need to look at. In this paper, it was a meta-analysis of many papers that were looking at how biomarkers could help identify patients with Alzheimer's disease as compared to controls. And they looked at whether there were some image modalities that were more accurate than others, but they also looked at how the images were analyzed. And what they conclude is that the accuracy of those biomarkers depends at least as much as in terms of how the images are analyzed than the images themselves. So the choices, the methodological choices that you make actually influence a lot your results. And yeah, I think it needs to be just very clear in our mind when we read papers about biomarkers and people being positive for something. And just, this is the last slide on technical aspects, I think. As I've told you, there's these stages looking at amyloid and degeneration. But usually people, when they try to identify people with neurodegeneration, they look at MRI and FDG PET, and we somehow consider that those biomarkers mean the same thing, or they can be used for the same thing. When actually, it's not that clear that they exactly represent the same. And here, we've shown before that atrophy and hypometabolism are correlated in patients, but there are slight differences. For example, atrophy is maximal in the hippocampus, and hypometabolism is more in the neocortex. And when you actually use those two tools to classify people on like whether they're positive or negative, they actually don't agree all the time. And you can see here that in 30% of cases, they're going to give inconsistent information. So what do you do with those patients? And when I was trying to explain this to a student, I actually used a metaphor with cognitive tests again. For example, if you try to, if you want to identify executive dysfunction in patients, you can use multiple tests. And those are, I think, among the most classical. So I'm not talking about the Wisconsin sorting cars. Sorry. And of course, no, this is unpublished data from our little cohort of patients. But of course, patients would tend to have low score on the Stroop task, have low score on the TMT task. They are somehow correlated. But if you want to classify people as positive or negative, you can use this classical threshold of minus 1.65 z-score. And you can see patients that are consistent in like no deficits or deficits, but there's also patients with like inconsistent biomarkers, not biomarkers, cognitive tests. And I think this is an illustration of what we can have with biomarkers too. So maybe it's more meaningful to think about those tests to understand that biomarkers that are supposed to assess somehow similar things might disagree. So I'm just going to go through a few studies that looked at those people that are positive on biomarkers. And this is one of the first study that was a big one on those staging. And they showed that actually almost 50% of the elders in their study had no positive biomarkers. And you can see that a lot of patients fit in their categories, but actually it doesn't, if you add them, it doesn't go to 100%, because actually at least a quarter of those patients had only neurodegeneration and not amyloid. So they don't fit in that model of Alzheimer's disease progression. And they were called SNAP for a suspected non-Alzheimer's disease pathology. And they've been the focus of intensive research because they were actually not that expected. And they don't seem to be just an artifact because they've been reported very consistently in many different studies using different populations. And the interesting thing about those patients is that they don't seem to be very specific on anything. At least in those studies, they didn't have more white matter lesion, they didn't have more vascular risk factor. They're very, very unspecific, except that they have a low proportion of ApoE4 carriers. And as you might know, ApoE4 is the main risk factor for Alzheimer's disease, the main genetic risk factor. So this is another representation of those healthy people with Alzheimer's biomarker. And it's actually a huge sample, population-based sample from the Mayo Clinic in the US. And here you can see that age is a major determinant of those biomarkers in healthy controls. And from the age of 73, 75, at least 1 out of 2 people have at least one positive biomarkers. So first thing is that those biomarkers are very frequent in the cognitively normal. And I just wanted here to show you that ApoE4 is also influences the probability of biomarkers, and especially the amyloid biomarkers here. So you have people without the ApoE4 allele, and here the influence of ApoE4, you can see it on the green curve that show people with amyloid deposition. I'm going to go over this real quick and switch to the cognitive correlates of those biomarkers, because you can expect that if people have something in their brain that suggests Alzheimer's disease pathology, you might expect that they have some cognitive or at least subtle cognitive difficulties. It's not that simple to talk about cognitive correlates, because it depends on whether you look at cross-sectional correlation, longitudinal correlation, and the literature is pretty confusing on this topic. I just want to show you that those relationships between biomarkers and cognition are not that clear or that strong. And here you can see a lot of studies that showed an effect of amyloid deposition on memory, for example, here. So in this study there was an effect on memory but not executive function. In many studies there was no effect at all of amyloid. So people had this amyloid in their brain, but their cognition seemed to be pretty normal. And you also have intriguing studies showing that people with amyloid in their brains had better cognition. Here, I think in this paper it was better episodic memory and higher fluency. So those results are less clear than we thought in the beginning. And this is a meta-analysis of those studies looking at correlation between amyloid and cognition. And the only significant correlation was with memory, but you can see here the correlation coefficient is extremely low. So those links are very subtle. And in addition, they seem to be influenced by a lot of different factors. This was a first result that was, I think, a little bit surprising. What is different, though, is when you look at people with biomarkers and you follow them up in time, and you can see that people with a lot of positive biomarkers tend to develop Alzheimer's disease or cognitive impairment more than people with no biomarkers. And when you and there's also studies showing that, well, insisting on the fact that having one abnormal biomarkers does not changes a lot, but when you have multiple positive biomarkers, then your cognition is really expected to decrease over time in the subsequent time. One last thing on people with so I've mentioned the effect of biomarkers on cognition using cognitive tests, as we basically do most of the time. But there's also this new focus on subjective cognitive complaint, which is the idea that people complain about their cognition when their cognitive tests are normal. And it's been shown many times that those people with subjective concern are at higher risk of developing Alzheimer's disease. And what's interesting is that those people, when compared to people without concerns, they have atrophy in the middle temporal lobe, they have hypometabolism, and they have beta amyloid plaques. So they have all positive biomarkers as a group again. And both predictors are, both biomarkers, amyloid and neurodegeneration, are predictors of those subjective concern here. So I guess I got like 2 minutes. Okay. So there have been a lot of studies on biomarkers, but there's still a lot of work to do on those, especially because as I've told you, a lot of techniques differ between labs, and we basically, each lab usually use their own technique. So there have been a lot of efforts to harmonize image analysis, and this is an example on hippocampal volume. And there's also the idea that we could use new techniques to optimize the use of those images. And for example, this is a study we've done in Caen, developing a new MRI sequence to identify subtle differences in the anatomy of the hippocampus. And when you look at specific subparts of the hippocampus, like in blue, the CA1 subfield, you might get a better ability to capture Alzheimer's disease. And here you can see the hippocampus in MCI was only decreased by 12%. And when you look at this blue area, the CA1, then the effect is really more important. And then using this specific volume, you get better sensitive techniques. And also from a more fundamental point of view, we were able to show that this CA1 subfield actually really correlates with encoding deficits in episodic memory. So using those patients, we can also learn a little bit more about the specific brain correlates of different memory subsystems. I'm a little bit late, so I'm going to go quick on this. I just wanted to show that we've got this idea that environment and lifestyle can influence cognition, and then can influence maybe how pathology influence cognition. But there's some new data showing that maybe lifestyle and environment could influence pathology itself. And there's nice data on using biomarkers like here, showing that elder people with a high lifelong cognitive activity tend to have less amyloid than people with low cognitive activity. And this has been also replicated with physical activity. Here you can see more amyloid deposition in people with a low physical activity. And there's also some data on the role of sleep. So basically, a few take-home messages. Those biomarker abnormalities, they are not that easy and not that straightforward to highlight in the brain. They can be frequently abnormal in cognitively normal elders. And in those people, they're not that related to cognitive deficits, except when you looked at subjective feelings of cognitive deficits and subsequent decline in cognition. And of course, the more biomarkers you have, the more positive biomarkers you have, the more at risk you are of cognitive decline. The other thing I want to insist on is that those different biomarkers, they don't mean the same thing, and they're not redundant. One important point is that neurodegenerative biomarkers can be found without amyloid, and that's not from a very clinical perspective, but more from a fundamental point of view. When we try to understand what causes Alzheimer's disease, this was not expected. And of course, those biomarkers are very promising for research purposes, but for clinics at the moment, they are not very useful yet. So I'd like to thank you for your attention and thank all the people from the lab in Caen who helped me with this presentation.